Hey there, and welcome to This Explainer. Today we're cracking open a seriously fascinating chapter on biotechnology. We're taking these concepts completely out of the textbook and seeing how they're actually being deployed in the real world to solve some of humanity's absolute grandest challenges. We're talking about everything from figuring out how to feed billions of people to literally rewriting the genetic code to cure diseases that used to be totally untreatable. It's an incredible story of human ingenuity, and I am so pumped to dive into these insights with you.
Okay, so let's ground ourselves with a quick recap of what biotechnology actually is. We aren't just talking about sterile, isolated lab work here. We're talking about the industrial-scale production of biopharmaceuticals and biologicals. Basically, we're using genetically modified microbes, fungi, plants, and animals as living, breathing factories to create solutions that directly improve human life. This spans everything from therapeutics and diagnostics all the way to bioremediation and energy production.
Now, to make these massive biological solutions a reality, scientists really focus on three critical research areas. Think of it kind of like a high-performance engine. First, you absolutely need the best catalyst, which is usually a highly tweaked microbe or a pure enzyme. That's your engine. Second, you have to engineer the optimal environmental conditions for that engine to run at peak efficiency. And third, once your biological factory churns out what you want, you need advanced downstream processing technologies to purify the final protein or organic compound. Those three pillars are really the whole shebang when it comes to making modern biotechnology possible. So, here's our roadmap for this explainer. We'll start with what biotechnology is, then move into cultivating better crops, check out the GMO pest defense, look at engineering modern medicine, and finally, wrap up with curing genetic defects.
Section 1. Cultivating better crops. The agricultural challenge.
So, how do we ensure there's actually enough food for a rapidly expanding global population? Well, while the Green Revolution was huge and tripled the global food supply, it still wasn't enough. We generally have three options for bumping up food production. Number one is agrochemical-based agriculture, which leans heavily on fertilizers and pesticides. But honestly, for farmers in the developing world, those chemicals are often way too expensive, not to mention the heavy environmental toll they take. Number two is organic agriculture. But organic farming has some pretty strict yield limitations. It simply can't produce the sheer volume needed to feed billions. Which leaves us with number three, genetically engineered crop-based agriculture. Traditional breeding techniques just couldn't keep pace with demand, so engineering crops right down at the genetic level became a completely necessary leap forward.
But even before full-on genetic modification, scientists came up with this incredibly elegant solution back in the 1950s called tissue culture. They figured out totipotency, which is basically a cell's superpower to generate an entirely new whole plant from just a single cell or a tiny plant part called an explant. By popping this explant into a sterile, nutrient-rich mix containing things like sucrose, vitamins, and growth regulators, you get micropropagation. This lets us rapidly churn out thousands of healthy, genetically identical plants, known as somaclones, in almost no time at all. In fact, if you've grabbed tomatoes, bananas, or apples at the grocery store lately, there's a really good chance you're eating the direct result of this exact science.
Section 2. The GMO Pest Defense, Engineering Solutions.
Tissue culture is amazing for propagating plants, sure, but how do we protect those crops when they're out in the real world, facing drought, disease, or hungry insects? That's where we get into genetically modified organisms, or GMOs, and the perks here go way beyond just getting higher yields. We're talking about tailoring crops to have increased tolerance to abiotic stresses, meaning they can shrug off brutal cold, drought, or extra salty soil. They also drastically cut down our need for toxic chemical pesticides. This isn't just a neat lab trick. It matters immensely. It prevents massive post-harvest food losses, makes plants use soil minerals way more efficiently, and can even pump up nutritional value to fight global health crises. A great example is golden rice, which is specifically enriched with vitamin A to help prevent childhood blindness.
Just take a look at this striking visual proof of the technology in action. Over on the left, there's a cotton boll that has been absolutely decimated by bollworms. It's totally ruined. But right next to it, you see a fully mature, pristine cotton boll. This healthy plant was genetically modified to produce a specific protein called Bt toxin, which acts as its very own built-in biological armor against the pest.
So, how does this built-in armor actually work? It is a total masterclass in targeted defense. The gene itself comes from a bacterium called Bacillus thuringiensis. Now, the toxin hangs out in the plant as an inactive prototoxin, which is great because it means it doesn't kill the bacteria or the plant itself. But here is the really fascinating part. When a pest insect takes a bite,
The specific alkaline pH of that insect's own gut dissolves the crystals and activates the toxin. It's wild. The bug's own stomach ironically triggers its doom. The activated toxin binds to the insect's midgut cells, creating little pores that cause the cells to swell up, burst, and ultimately kill the insect. It's a biopesticide that's perfectly specifically tailored to the pest biology.
And here is another stark contrast, showing off a different biological defense mechanism. On the left, we're looking at the roots of a typical control plant that's heavily infected with parasitic nematodes. You can see they are just covered in those damaging root knots. But on the right, we have transgenic roots 5 days after they were deliberately infected by the exact same nematode, and yet they are completely clean, healthy, and protected.
The reason the plant on the right survived is because of a totally novel defense strategy called RNA interference, or RNAi. Think of this as a cellular mute button. Using agrobacterium vectors, scientists introduce nematode-specific genes right into the host plant. The plant then naturally produces both sense and antisense RNA, which bind together to form double-stranded RNA. When the nematode tries to feed on it, this double-stranded RNA kicks off a process that literally hits mute on the specific messenger RNA of the parasite. It completely stops the parasite from producing the proteins it needs to survive. The pest just cannot live in the transgenic host.
Section 3. Engineering Modern Medicine. From Symptoms to Solutions. Let's step out of the agricultural fields for a minute and see how all this translates to the medical frontier. Because if you or anyone you know manages diabetes, this is the exact technology that changed everything. Back in the day, insulin had to be painstakingly extracted from the pancreases of slaughtered cattle and pigs. Because it came from an animal source, it often caused patients to develop severe allergies and really unwanted immune responses. But today, we use recombinant human insulin. By turning bacteria into microscopic factories, we can mass-produce insulin that is perfectly 100% identical to the human hormone. It's incredibly scalable, highly effective, and completely safe.
Now, actually getting bacteria to create human insulin wasn't just a walk in the park. In mammals, insulin is synthesized as a prohormone. Notice how the proinsulin has this extra stretch of amino acids called the C-peptide. You can kind of think of it as a safety cap. For the insulin to become mature and actually do its job in the body, this C-peptide has to be snipped off and removed, leaving just the A-peptide and B-peptide linked together. Trying to get bacteria to assemble this whole complex structure properly was a massive headache for scientists.
But then came the massive breakthrough in 1983 by the American company Eli Lilly. Instead of trying to force the bacteria to process that complicated proinsulin, they performed a totally brilliant sidestep. They prepared specific DNA sequences corresponding separately to just the A chain and just the B chain of human insulin. They dropped these sequences into the plasmids of E. coli bacteria so they would produce the two chains completely separately from one another. Then, they simply extracted them and linked them up in the lab using disulfide bonds to form perfect, mature human insulin. It was literally a biological heist, and it completely revolutionized medicine.
Section 4. Curing Genetic Defects. The Promise of Gene Therapy. We've seen how biotech treats symptoms, but what if we could actually go in and correct the very code of life? That brings us to gene therapy, which allows us to fix a genetic defect diagnosed in a child or even an embryo. Take adenosine deaminase, or ADA deficiency, for example. It's a severe disorder where a crucial immune system gene is just straight up missing. In the past, doctors tried treating this with bone marrow transplants or frequent enzyme replacement injections, but neither of those are actual cures. Then, in 1990, a massive historic milestone happened. A 4-year-old girl with ADA deficiency received the very first clinical gene therapy. Rather than just managing our symptoms, doctors delivered a normal, fully functional gene directly into her cells to permanently compensate for the broken one.
And that really brings us to a remarkable realization, doesn't it? We've journeyed from using tissue culture to clone crops, to engineering cellular mute buttons that stop pests in their tracks, to turning everyday bacteria into life-saving insulin factories, and finally to rewriting human DNA to cure genetic disorders. Biotechnology really isn't science fiction anymore. It is the absolute vital tool set of our modern world. So, as we keep mastering this ability to rewrite the code of life itself, what other grand global challenges are we going to solve next? I'll leave you to ponder that. Thanks so much for joining me for this explainer.